but we're just really interested in the live sponges today.
Than it actually was.
But that doesn't mean that dead sponge doesn't provide habitat to a number of different animals. We've definitely seen some very large dead sponge masses that have supported, you know, brittle stars, lobsters, shrimps, and crabs within the folds of the sponge. So even after their passing, corals and sponges are definitely part of the habitat in this area and in other seamount and canyon areas.
Alarming that I can tell from sonar.
Absolutely, that sponge branch was almost more conspicuous for its lack of apparent associated organisms than it was for having them. We almost always see them with it.
So the only thing
the squat lobster is looking pretty squirrely here. We'll see if he's going to make his escape as Deep Discoverer approaches.
That much longer. So the ridge is going kind of like this.
It might, but you know, it might not.
Purposefully kind of going kitty corner. Okay.
Yeah.
I'll keep it at 260 then.
If we landed right in front of it, it definitely would make its move.
I'll put it in.
Well, and also that when the dead sponge happens to land on an area of soft sediment like this, it doesn't make as good a habitat as if it was on top of a rock or a place that might not move.
So, pivoting back on something that I was saying a couple minutes ago, this is the Deep Connections expedition, and one of the connections that we're really interested in are links between the deep sea environment and the continental shelf. And the continental shelf is generally thought to be in the area of about 120 meters below sea level or shallower, while the deep sea depths range from thousands of meters, 1,000 meters down to 6,000 meters or even more in the trenches. These features submarine canyons, which are canyons carved within the seabed, as well as seamounts, which are these protrusions that are formed from mantle hotspots pushing through the lithosphere, serve as bridge communities between that shallow continental shelf and the deep sea. They do so in a number of ways, biologically, chemically, physical oceanographic, as well as sediment conduits. So they're really important to study from various disciplines. They mean different things to different scientists, but they're important for them all to study. And one thing that's really apparent from the seabed that we're seeing right now, which has transitioned into predominantly fine-grained sediment, is these really distinctive sediment waves. And at these grain size, these grains are relatively small and could probably be picked up by currents that aren't extremely strong. This is probably evidence that we're seeing right now for currents probably stronger than are active in the area right now. This seems pretty serene, so this might be something like slack tide in this area would be the terrestrial analog that we would use. But at some point there are currents moving above the seabed and organizing it into the wave field that we're seeing right now.
Seamounts and canyons are often considered biodiversity hotspots in the deep sea. That means that we see a wide range of animals that are very densely packed in these small, relatively small areas of the deep ocean. And that's because these areas have similar features within them, being that canyons have steep canyon walls and these seamounts have steep slopes. And these types of steep and rugged terrain are really important for corals and sponges and their associated community of organisms to live. So what we're looking at right now is a halosaur. It's a type of eel-like fish, and it's floating vertically in the water column. I say eel-like because it has this long tapering tail and swims in a very efficient motion by undulating that tail in an eel-like manner.
Sorry,
repeat
your last? I tried the maximizer.
So I really like halosaurs because they have these really adorable pectoral fins that are tilted upward, almost like little wings. So they look like dragons in the deep sea, at least to me.
It does. I'll definitely agree with that. And I can't remember who it was. One of our shore-based scientists was elaborating a little bit on why they have this really distinctive vertical position. And it's for two reasons. It makes it efficient for it to prey on
down to the bottom of it and then
organisms in the water column above it, and it's also for any potential predators. It produces a really small footprint in the water column if you're viewing it from up and down, which is probably the way that you would be seeing it in the pelagic environment. It makes it just kind of less obvious and less conspicuous if you're looking down and you only see a little dot instead of the full length of this fish.
That's particularly true in the water column.
So just go ahead and put another one in,
2 days from now, we will be doing some water column explorations, and the animals
like
living in the water column, some areas between the bottom and the surface of the ocean, is one of the largest ecosystems on our planet, and it's also one of the few ecosystems that we know the least about. So we'll be transecting across these areas at different depths and seeing what kind of animals live there. And like Jeff was saying, a lot of animals will adopt this vertical swimming habit, and they'll have very, very large eyes in order to pick up light coming down from the surface above, because at this depth we won't actually see any light at all, but in shallower waters some light does penetrate, so they have these large eyes that will capture the fewest photons possible and try to resolve an image. And by looking upward, they can see anything that's making a profile above them, and by being vertical and reducing their profile, any animals below that are looking for a tasty snack are less likely to see them. So it's a really interesting thing. Oh, there's a tube anemone. Can we take a look at that down at the bottom of the screen?
Sure.
So tube anemones are pretty cool too. They create these sort of paper-like tubes within the sediment that they actually live inside. And one way to identify this as a tube anemone is that it has a ring of tentacles along the outside of this anemone disc, and then they have a second set of tentacles around the mouth of the organism that are much smaller.
Go ahead and come in video.
So if you just give it a few seconds, we're going to take a zoom in on this tube anemone, and you'll be able to see that second row of tentacles right around the mouth. So the mouth is located straight in the center of this organism. Or it will sense us in the water and zoop, back into its tube.
Yeah, thankfully it didn't close up shop entirely, and we can still see it. They're a little hard to see, but I can definitely see that distinctive inner ring of tentacles that you were talking about closer to the mouth.
Yeah, these animals can be particularly difficult to sample because they will pull into their tubes, and so studying them can be a challenge. Also, being able to see the different structures within the animal after it's pulled into the tube makes it very, very difficult for taxonomists to see. So this wouldn't be something that we would attempt to collect. We would have to either try to take a core of it, would probably be one of the better ways to collect this without disturbing the organism itself. And then once we get in the lab, you'd have to relax it so the tentacles would come out, and all those internal spaces within the anemone that the taxonomists use as a way to identify it to species would also be relaxed so that they could see those different structures. But as we looked at it really closely, people like Tina Molodtsova, who know a lot about these animals, she could probably come to a conclusion as to what species this is based on where we're seeing it, the depth, and the location.
Pilot, can we focus in on those sponges to the left?
Yeah, on the left side, we're definitely seeing some more dead sponge. So I'm thinking like right on the top of this ridge that we're making our way up, we'll probably see a lot of really large sponges, or at least there were a lot of really large sponges sometime in the past.
Yeah, and Megan, what I'm interested in, what I'm wondering is whether these sponges were anchored to something here in place, if this was similar to that debris field that we've seen before, and based on the ripples that we're seeing on the seabed, is whether there's been a large amount of sediment transported over this area that kind of buried everything that was growing on a previously more rugose and rubbly field that it preferred, and then these waves of sediment moved and migrated over this area and kind of smothered everything here. That's a working hypothesis.
Yeah, that could happen. I know you do a lot of work with submarine landslides and understanding the movements of rocks and sediment within canyons. And I would assume that similar types of movements happen along the sides of seamounts.
Yeah, that's right. And if this is the case, what I'm postulating, that would be less about seabed instability, which is generally thought to be the movement of the seafloor itself, and more about more superficial sediment transport, which would be individual grains being picked up and moved across the sea. And you're absolutely right about the timescale comment. Some of my colleagues at the Naval Research Lab use particle image velocimetry to observe the movement of sediment grains that would create these kind of ripples that we're seeing on the seabed. When me, who studies seabed instability and submarine landslides, if I plopped down here and waited to observe a submarine landslide happen in real time, depending on the location, it could be the end of my lifetime before that happened. So definitely different time scales that we're talking about here. So right now, as we've mentioned a couple times before, we are in the Northeast Canyons and Seamounts Marine National Monument. And this is two distinct separate areas of the seafloor that have been protected in terms of certain fishing activities. This was established in 2016. We are a little before the 3-year anniversary of that right now. And it's separated into two different units, one of which the Submarine Canyons unit, which we were exploring right before we had to head back into shore for Dorian, and the Seamounts unit, which is where we're at right now, which is where Bear Seamount is within. And these national monuments are relatively important. They harbor a lot of really diverse deep-sea communities, these biodiversity hotspots that you were talking about a couple minutes ago, and protecting and managing these resources, at least having some areas that are protected from various fishing activities and can be destructive for these really slow-growing and fragile deep-sea communities is important for the preservation of biodiversity in the future, as well as for effective fisheries management.
And one of the most important things about this managed area, this Marine National Monument,
ooh, octopus.
The Northeast Canyons and Seamounts Marine National Monument is important because they act as as a living laboratory, and our explorations in this area can teach us more about how areas of the ocean are connected, how the Seamounts section and the Canyons unit are connected, and now I'm distracted by this awesome octopus.
It's a cephalopod and it's so cool.
I love how it's walking across the bottom. It's just out for a stroll. It's a beautiful day to be on a seamount.
How would you describe this motion, Megan? It's
It's just a a deep sea stroll through the octopus's garden.
Yeah. So, do we have any specifics on what type of octopus this is?
I believe it's the same type of octopus you saw when I was at lunch that day, and I totally missed it. Granulone. I'm not going to try to remember how to pronounce that name. Starts with a G.
Okay, if you'd like to take some more video, I think I got a relatively stable platform. Come down a little.
This octopus is making my day.
I was just about to comment on the relative lack of mobile fauna that we've been seeing so far in this dive, other than a couple squat lobsters, and then our favorite cephalopod comes along to. Oh, I'm sorry. We had one last announcement. Hopefully, one last. Yeah, and it's really interesting as well. The just kind of rippling effect around its mantle, that kind of webbing between its tentacles. I haven't really seen or noticed that before.
Can you just keep an
and
eye
I'm
on
wondering,
it
as it moves by these brittle stars, I'm guessing it's just transiting right now. It's probably a little wary right now, given Deep Discoverer is staring it down with all its bright lights and all. But I'm glad it hasn't decided to enact a defensive posture in which we wouldn't be able to see this really distinctive squid swagger across the seafloor. Octopus swagger, my bad.
So octopus tend to be pretty curious in nature, and they will often interact. And
do you want me to follow
this one
video?
is
Do you want me to let
definitely
it go? What do you
swaggering as it's making its way across the bottom,
I
strutting
think we
its
can
stuff.
let him go.
Copy that.
It was good to see you, little octopus. Keep on keeping on.
Turn ahead. I don't know.
Is it that later in color? It looks like a large sponge, doesn't it?
I think it's just beyond. It's like 15 meters out.
Copy.
Okay, I finally found where I wrote down the name of
do I have enough
this
tether
octopus.
to get to that lighter
it's
color
in the genus Granulodone.
I'll hit it there.
And we saw one a couple dives ago
It's
at
a
the
big fish
Northeast
on the
Channel,
left.
incubating
Fish on
some
the left.
eggs on the underside of a rock.
In the water column, or?
And that one was had no interest whatsoever in moving. It was taking that posture both to protect the eggs against predators as well as to circulate oxygen-laden water
He's
through
swimming away from
the eggs.
us.
A position at which the female octopus will apparently maintain until its death and the hatching of the eggs, which is sad but also
I don't think we ought
very
to chase
dedicated.
him, but thank you.
Copy. Any interest in this sponge?
Yeah, it looks like a really interesting shelfy sponge that we're seeing here right now. This might be a living example of one of the ones that we saw downslope earlier on this outcropping of rock. I think we're going to sit down and take a closer look at this and all its many associates that we can see even from this distance.
Given the size of this sponge and how similar it looks to a lot of the pieces of sponge that we've seen, scattered about this dive,
yeah.
there might be
looks
more
like
of
one in
these coming up as we transit upslope. So I'm getting a good look at this sponge, and I'm trying to decide what type of sponge it is. It strikes me as being a glass sponge, likely in the family Rosellidea.
Nothing comes to mind.
But it's sort of more of a plate-like instead of a vase-like sponge.
Flow, but
what would you like to focus on video? Just right here? Or?
Yeah, just on the sponge right where it is,
Yeah,
so you
it's
can
really
stay
neat
put.
to see the tendrils, what looks like, what I'm assuming are the brittle star on the other side of the fold of that sponge peering over the top, and you can see why the edges of these sponges are such coveted real estate among the associates, because it gets it right directly perpendicular to the water flow that's coming through and exposes it to the best food in the water column.
Video's clear.
Copy that. Feel free to come full wide, and we'll continue forward.
And it's hard to know if what we're seeing right now is an outcropping of the actual seamount ridge arm itself, or whether it is a clast or boulder that had failed off the wall of the seamount and was deposited later. It's a little hard to interpret a geological setting with this small view.
Yeah, it looks like there might be a little bit of relief up ahead.
I mean, I would hazard a guess that this is actually bedrock from just like a part of this seamount. And if it was a bit younger, we might be able to make out the different pillow lavas that made up this part of the seamount. And if we are lucky, we might actually see some pillow lava formation.
It's in the same direction.
And when I say pillow lava, it's a way that the lava is extruded from
another move.
How
do you feel about another
the
20
volcano.
meters, 260?
So pillow lavas are formed relatively slowly. It starts off breaking out from the main lava tube, and like a tube of toothpaste is slowly extruded and creates this very lobate formation. When the lava is extruded fast, it tends to form a sheet flow. And as we get this sort of a side look at this part of the rock, it looks like there's a lot of layers. And you notice those types of layers in some layered lava flows, because these flows happen over many, many years at different times. And so they could be layered just like you would see the layering of a sedimentary rock. And looking at those many layers, you could eventually figure out when different lava flows occurred, because obviously things on top happened more recently than things on the bottom. Another thing we can be seeing as we're seeing layers is the layer of manganese crust formed on top of the basalt. And so manganese crust happens on top of basalt at very, very slow rate, and it thickens over time. And that's my knowledge of geology for the day.
It's pretty good, Megan, and even though I am ostensibly your geology lead on this cruise, I think your knowledge of igneous geology is probably a little further than mine is, which goes along with Megan's expertise and experience working with the Pacific environment, a lot of volcanic environments, which I don't have too much familiarity with.
So right now we've zoomed in on this lemon yellow sponge. I believe this sponge is a type of Hertwiggia glass sponge. We made a collection of one of these sponges a few dives ago, and I thought it was really interesting because we collected something similar that was purely white. And so it's going to be very important to sponge biologists who are trying to figure out the genetics and the connections between different sponges across the Atlantic. And one of the main questions is this yellow Hertwiggia sponge the same as the white one? Do they have different color morphs, or are they separate species?
Yeah, I think I was pushing down too hard and
And
started
you'll
rocking
notice
myself.
that part of that sponge is dead. It's that brown, sort of granulated dead sponge look. And this sponge here in front, the sort of really crazy convoluted sponge, we keep seeing that one. It's not one of the ones I'm familiar with, and it looks very similar to the unknown sponge that our Canadian partners supplied an image to. Another thing I'm seeing right now is we're getting a side view of this, trying to look at its attachment to the rock. It looks like it's actually all coming together in more of a vase attaching to the rock at one single point. And as we zoom in, I'm seeing more of this like kind of crisscrossing
We can
road
hit it with the fog lights
as part of the framework of the sponge.
If you don't mind.
So I believe this is a sponge in the family Euplectellidae. It could be an Atlanticella, which forms this sort of base-like but very open type of sponge morphology. And as it gets older and larger, it starts to convolute and curl under and have a very interesting morphology for the entire sponge. But it might start off looking more like a vase. So that was actually a really good view because, you know, it was from the top view, it looked completely different. But having the side view was very helpful because now we know that it's probably a euplectellid sponge in the genus Atlanticella.
Video's clear.
Yeah, and one of our shore-based scientists, Nolan Barrett, confirms that his suspected identification of the sponge as well.
We also have images of a sponge very similar to this one within our deep sea animal guide, which you can find on our website at oceanexplorer.noaa.gov, or you can just Google Okeanos Animal Guide. It'll probably be one of the first couple links that you see, and as you click through the different images of sponges, you can see what I'm talking about when it comes to this Atlanticella sponge. So the guide is organized by phylum, which means these major groups that we've classified animals into, and that includes sponges, corals, which are cnidarians, sponges are porifera, echinoderms, which include all the sea stars, these feather stars that we're seeing, urchins, and sea cucumbers, and then there's the fishes, which are the chordates, and the arthropods, which includes all the crabs and shrimps that you'll be seeing throughout the dives. And it's all a drop-down menu. So as you click through, you can see different galleries, and these galleries feature many different types of animals.
Yeah, and I can't overstate how useful this identification guide has been on this expedition alone. I can't count the number of times that Megan has referred for that for a tentative identification.
Yeah. Yeah. All right, pilots, how do you feel about another 20 meters?
That other move.
I feel like I'm just kind of...
Pilot, can we zoom
one.
on that
right,
floating animal that just went off screen on the lower left-hand side?
Might have turned my head a little bit.
Oh, it might have floated away.
What was that? What leaded you? You're very quiet.
There was this floating animal coming up from the bottom.
Yep. I believe video is busy.
I think it might be a type of pearlfish. Yeah, so looking at this fish, it looks like it might belong to the family Liparidae. Those are the pearl fishes.
Am I guessing too much to assume that its name is related to its color?
I'm not sure if it has anything to do with the color, and I'm not sure where it gets its name. But these are the fish that in shallow waters have been quite sensationalized as the types of fish that live inside the anuses of sea cucumbers. Now that fish is not the type that lives there. It is a free-living fish, and Liparid observations in the deep sea are relatively
You think we could do 270 for one move and follow this ridge for a little bit?
Yeah, we can do that. Yeah. 20 meters at 270.
Sure.
All right, I'll put it in.
Welcome to the conference center. At any time during this message, please enter your passcode followed by the pound sign. Your passcode has been confirmed. If you need technical assistance during your call, press *0. After the tone, please state your name, followed by the pound sign.
Go ahead.
Thank you. You are the first caller to this conference. Please wait while others join.
Try that again, watch lead. As of now, they are still not functioning.
Science Watch leads, this is video. Thank you for your patience. We are back on the phone call and ready to zoom as desired.
Thanks, video. Okay, yeah, we are transiting over a fairly rugose terrain right now.
So you're going up slope.
If you've just joined us recently,
not go too much further
this
here.
is dive number 8 of EX1905 Leg 2, Deep Connections, Submarine Canyons, and Seamounts. We are on the latter half of this group in the Seamount area right now, Bear Seamount to be specific. Our Leg 2 got subsectioned into 2 smaller legs by Hurricane Dorian. But we are back out and excited to be checking out the seabed elevated features when previously we were looking at erosional submarine canyons for the rest of our expedition. So even though these environments are geographically different, morphologically different, ones are depressions and ones are elevated features above the seabed, they both kind of serve similar functions in terms of being biodiversity hotspots. A lot of the relief that comes associated with them, which are sheer walls incised in submarine canyons, as well as the exposed ridges and flanks of the seamounts that we're ascending up right now, provide a lot of suitable substrate for foundational organisms, sponges and corals that get all kinds of associated organisms along with them, just like we're seeing right now. And it looks like the associates with this sponge include the brittle stars that we've been seeing abundantly in this area. I think I saw a squat lobster as well to the upper left on this sponge. And even though the sponge is encrusted in sediment, you can tell by just that brilliant pearly white color that it's still living and still a fairly healthy specimen.
It could be heavy too. It is a relatively large sponge. So this sponge that we're looking at is probably another type of euplectellid sponge.
Up to the upper left.
So I was trying to get a look at the base. It kind of looks like it's coming together in a vase similar to that lenticella sponge. But I'm believing this one is a little bit different. And that's because we're seeing this very regular dictyonal framework. That's that very crisscrossing pattern of spicules that you're seeing within the walls of this sponge.
Feel free to come
so
out
these
when
corals
you're ready.
and sponges that we've been seeing along Bear Seamount are often considered ecosystem, or like, what was I trying to say?
Foundational
here
organisms?
for the next few minutes because we're going to do a shift change, so we might just wait.
They construct they construct as they grow, they become more complex, and the complex structure of these organisms is really important to a bunch of species, like you were saying. And corals and sponges can provide a number of different functions to associated animals, like providing shelter from predators, providing areas for breeding, spawning, and nursery habitat for a variety of different types of animals, which is very important to the biodiversity of the deep sea.
Yeah, Bob Carney, one of our scientists on shore, describes them as engineer species, which I think that's a really apt term. I've never heard it before. I think it's referring to the same thing that in the coastal analog to this environment is usually referred to as a foundational species, Spartina alterniflora, the marsh cordgrass, which grows in huge abundance in the coastal environments in a lot of different areas wherever there are salt marsh, generally speaking. And they grow in such density, and they provide a lot of relief and surface area to be colonized above the surface of the marsh, and they get used and do important ecosystem work in terms of providing substrate for some organisms to live their entire lives on, and sometimes larval species,
in on that,
juveniles, hiding areas before they move on to a different habitat. But the corals and sponges that we've been seeing encrusting the rocks in this area, rocks and sediment in this area, serve a similar function in a completely different ecosystem, very far removed from the coastal marshes that I have a lot more experience with.
Copy that. Who's on the next shift? Are you on? Okay. Yeah, we can. So do we need to? Well, you want to come up for a minute? Copy that.
Another thought that might come to mind when you look at Bear Seamount, we were looking at the bathymetry of Bear Seamount, which was mapped during an earlier leg of the Okeanos expedition, is the shape of it. It looks really flat-topped, and that actually forms it into a different subset of seamounts, which I believe I've been mispronouncing my entire life up until this morning. I believe the correct pronunciation is Guyo. Guyot. There we go. It's spelled G-U-Y-O-T. And a guyot is what happens when a seamount either extends up into the water column, or if the hotspot occupies underneath it long enough, breaches the surface and it becomes an island, as you might, as Megan was saying earlier, but be familiar with the Hawaiian Islands. Over time, a combination of wave erosion as well as thermal subsidence,
you, Workshop?
which is just the gradual cooling and contraction of a material, results in it becoming more flat top from the wave erosion, as well as closer to the seabed and losing elevation or decreasing depth, getting closer to the seafloor over the course of millions and millions of years. As we mentioned, the approximate age of this seamount that we're surveying right now is 100 million
I'm just wondering
years
if we need
before
to
present. So there's been a huge range of modifications that have occurred to it since it was being extruded from the seabed.
Okay, copy that. Thanks. Copy that.
Another interesting thing about seamounts and canyons is that there are these relatively isolated areas within the deep sea that have these high concentrations of life, high biodiversity, and high density of different types of animals. And we're seeing these animals here for almost the same reasons. Current flow around seamounts and current flow through canyons make for really good areas for these animals to inhabit, because high current flow brings food to these animals. It wipes away sediment that might cover hard substrate, providing more area for animals like corals and sponges to colonize. And with the more food in an area, the more animals can inhabit there, and these animals, corals and sponges, are ecosystem engineers building up the rugged structure of the walls of canyons or the flanks of seamounts, making them more complex and providing more habitat for various animals.
Yeah, and it looks like to the top center of our screen right now, we can see a sponge that looks fairly familiar. It looks similar to a couple specimens that we collected, one of which was our very first dive in the Gully Canyon Marine Protected Area in Canadian waters, and one more recently, I believe potentially in Kinlan Canyon, which had a similar morphological shape, but a different texture that was identified it as a different species.
You remember correctly, Jeff. So this one is a much larger specimen than the two that we collected previously. The first within Gully Canyon, which was in the marine protected area in Canadian waters, and then this one looks more like the one we collected in Kinlan Canyon.
The ship's not moving.
It has more of a bumpy outside texture, and as our pilots get situated here, they're going through a change of shift. We'll probably make our way over there and look at the framework of this sponge and try to see its texture. But from here, it looks like it seems a little more bumpy, more like what I would describe as Regadrella.
Copy.
If I were to consult my animal guide from the Pacific. But it could also be in the genus Dictyalis, which has more regular dictynal framework, more squared off, and regular small pores along the side of the sponge. Another interesting thing to look at with this sponge is the osculum, that top opening of the sponge. So these sponges often have netting that covers that sponge, and we also see associated organisms living within the sponge, namely these really cool shrimp. They live in pairs within the sponge, and we always just see two every time we've collected one of these types of sponges. And there has been a little bit of study about these associated shrimps, and they tend to always, the same species of shrimp tend to always be associated with the same species of sponge.
Before we call one in. Watch the pilot.
Go ahead, pilot.
Hey, do we want to take a closer look at this subject here?
Yes, please.
All right. Sorry, Nav, go ahead.
So this one's really interesting. It looks like in between, it looks like there's tiers of tissue as you go down through the center of this sponge. So I would assume that this particular specimen is much older than the ones that we collected that were also very small.
Let's come on in here.
It's hard to know exactly how old sponges are because there isn't a very good way to date them. Unlike, you know, corals that have hard skeletons that we can use different techniques to date.
And I definitely saw when we zoomed in, the osculum has that lacy covering. It certainly looks pretty similar to the one that we collected more recently than the Dictyalis from Gully Canyon.
We'll just get the general matrix of this.
Sounds good. And I'll just, on any of these shots, if you think
I also find it interesting to
help,
see that it's hard to see through the body of the sponge, even through some of these openings.
Thank you, Pilot.
Hey, Pilot, once we stop with the zoom in, can we get a look down the top of the sponge? We just want to see if there's some shrimp inside of it.
Sure, yeah, we'll try for that. It's kind of at a weird angle.
Okay.
But we'll try to look down as much as we can, yeah.
Yep, if not, no big deal.
Okay.
You can go ahead and do that, Pilot.
Okay, sounds good. Coming up.
Another cool thing about this sponge is it looks like it's attached to a small cobble on the seabed, and it probably at some point fell over, and then it sort of started growing back upwards again. So it kind of acted like a tree in a storm that got knocked over and continued to grow. These sponges, and also I've seen this with corals, have continued on in their life, and so that means that there's probably some pretty good current bring food to this animal down here.
Yeah, and the shape now is more reminiscent of a Viking war horn, actually, is what it looks like to me.
And since this has a relatively large osculum, as we look down into the sponge, we might be able to see if there are any associated shrimps living inside the sponge. Now, these shrimps, when they get into the sponge, they're larvae, they're small, and then they grow up and they can never leave the sponge. They will always live here together.
All right, video, that's it on our tilt.
Association.
So yeah, come on in.
So the sponge is providing a home for these shrimps that live inside there. I think I am seeing something in there.
This might be one that we hit with the fog lights.
Yeah.
Okay. Yeah, let's go
there's
for
a shrimp in there.
Coming on.
Yeah, thanks, co-pilot.
Oh, you gotta love that zoom. Okay, now I see it. It looks like the shrimp that we're seeing in view is a female carrying eggs. Oh, and I can see the eyes of the other. That's going to be the male. So it's always a female and a male, which is really interesting.
Get the other one.
But also not unexpected.
Sorry, Emily.
It's probably because when other shrimps try to make this their home, the first residents have say on who gets to live there. And so it's advantageous to have a male and a female so that they can reproduce. But I don't know if these shrimp like to share. Sometimes your apartment sponge is only big enough for two.
Translucent, isn't it?
But looking at these shrimp, they look a little bit different from the shrimp that we saw in the other sponge. They might be in the same genus, but the body of the others were a different color.
I'm going to pull out just a little bit.
So that main part of the carapace, and that main part of the carapace looked really inflated, and these look a little bit more regular. The only way to really tell for sure would be to make a collection, but since we already have two of these sponges, we're going to leave it at that.
If you're clear video, we can come out.
Video's clear.
Okay, should I take
but we did get some good visuals, and with some good stills of those shrimps, we can compare them to the ones that were collected throughout this expedition.
Absolutely, and it's really incredible the regularity at which whenever we see one of those sponges, it almost always seems to have those true shrimp associated with it. In a field where it just seems there's so much randomness and variance everywhere you look, it's kind of comforting to know that whenever we see one of those sponges, we're probably going to get a couple shrimp with it.
And you're right, Jeff. It's a really interesting association, and the more frequently we see it, that's sort of unexpected.
Yeah, let's start with 20 and then
how do the shrimp find the sponge? These are the really important questions that we're asking, because these sponges are not rare here, but it's not like you're seeing a sponge everywhere you look. And when the shrimp find the sponge, they're very small. So how do they know that that's the sponge they belong to? And if it's true that every single species of sponge has a different species of shrimp, how do the shrimps know which species of sponge is their sponge?
Looks like it's a little bit off to starboard on Sirius.
I agree, yeah.
Okay, I'm going to try and center up on it.
So that might come down to settling cues, and maybe there's something chemical between the shrimps and the sponges that the shrimps can recognize as larvae, and that's why they choose their particular sponges.
Okay. Yeah, let's get one in then. Yeah.
Yeah, that's what I'm working on now, Nav. And I'm rotating up a little bit, pilot.
Sounds good.
So I'd say roughly there.
And speaking
like
of larvae, you might be wondering how some of these sessile animals get here and how they can get to other places as well. So how do they get here really comes down to larval transport. So a coral on a seamount somewhere will reproduce, send their larvae up into the water column, and currents will bring those larvae to different locations across the ocean, and those larvae will hopefully be able to settle in a new location of hard substrate where they can be successful.
All right.
And that's how we can see how these different areas, these seamounts that are separated by many miles, and these canyons that are separated by many miles, are connected ecologically and genetically to one another.
Absolutely, and that's another link between the physical oceanography and the biology endemic in these areas is kind of whenever these larvae are transported, they're not doing that willingly. They're born with the currents. And this is one area that, as our mapping lead Mike White was talking before this dive, this is a convergence area where you have Arctic bottom water converging or moving over the Gulf Stream, which is warmer and saltier and moving to the north. And even though these water masses are moving over top of each other and below each other because they're separated out by density, there's some convergence at the boundaries, and wherever you get convergence there, you get turbulence, and a lot of times that results in settling out from the water column. So at that scale, as well as just the water mass in general being deflected and reoriented by the physical shape of the seamount, it can a lot of times create a lot of turbulence and eddies that resuspend some things and also create slack water in an otherwise moving flow that can cause sediment or biological larvae organisms to come out of suspension and say be born from one seamount and transported or advected to another one.
I can pay out a little bit and give you a little bit more.
Yeah, I think since we're headed that way, maybe we'll hit that next.
So water masses are identified based on their
toes here.
Their salinity, their density, and also where they're formed. So Arctic bottom water is actually relatively new
Emily, don't let me forget about that
a new water mass formed at the surface of the Arctic Ocean where ice formation is happening, and so that water tends to be very, very cold and very saline, and that's how it ends up at the bottom, and it moves slowly across the bottom of the Atlantic Ocean, eventually making its way through all the oceans. And as it makes its way, it does mix at these convergent zones with other water masses. I really like these sponges. These rosellid space sponges just seem to be their own little hotspot of echinoderms. So we keep seeing all these brittle stars and all these feather stars just clustered on top of these sponges. They definitely seem to be a good place to be.
I think
and I'm kind of wondering if
video's clear.
The brittle stars help keep the sponge clear of sediment. Like, they move their way across the sponge face, and they might be eating some of the particulates that get caught on the outside of this sponge. So if we were to touch this sponge, it would probably feel like a scratchy Christmas sweater. You know, it is made up of glass, so that's where you get the scratchiness from, but the spicules are very fine and they're densely packed, so it has more of a woolly appearance and a woolly feel. I think we spotted another cookie star resting on one of these small rocks. And now that this one is fully laid out flat, you can see why we keep calling them cookie stars. It looks like you took a cookie cutter and your sugar cookie dough and just cut out a piece, and it's this perfect star. This one looks very similar to the one that we were trying to get a good look at earlier. And you can see the scales along the outer edge of this star. And that can be a really good character for
I think that's a sea spider standing
which
above
genus that might belong to. By counting the scales and their size of the scales.
It back up. I'm trying to creep in. Are you close to max zoom there?
Yeah.
Okay. All right, let me get a little bit closer.
It is. So you're back. Good to hear.
Okay.
Is it? This looks like it might be the deeper water morph of Plinthaster dentatus, or it might be.
Okay, let's get another one in.
There was a species that you collected, or not you guys, but one of the previous expeditions collected.
Yeah,
that is a second species of the genus Sabogaster, which is something that I think is a new species collected on the prior dive. So we're still seeing undescribed species, even though we're working in these areas that are essentially in sort of the backyard of America's ocean habitats. It might also, we're at about 2,000 meters, so this might also be, wow. See, again, this is the difficulty with some of these really fine characters. How big is this? Do we have lasers?
Lasers are on. We just got to center it out. Can you come wide, Emily? I'm just
yeah, we're
right now.
Okay.
Yeah, we're zooming out. Yeah, so we can see it. There's our scale.
Right. Thank you.
So maybe 6 centimeters, I would guess.
Copilot, when was the first move?
It's relatively small.
Okay.
Okay. Yeah. So this could also be a sea star called Latonitaster, which is a goniasterid that occurs off. Generally speaking, the occurrence data are for something farther south. But
still moving for just a little bit more off that first one.
Yeah, see how the plates on the top are smooth.
Just about concluded, though.
Oh, wow. Well, I hate to be bold, but I was wondering if this is something that could be collected.
I'm sorry to say that we won't be able to make any collections currently. We're having little trouble with our hydraulic systems right now.
Oh, I'm afraid I missed something earlier about that. Okay, well, that's fine. There are a number of possibilities, and it's one of those things that if I had it in my hand, it wouldn't be quite so difficult. But there are maybe 3 or 4 possibilities. Again, it could be one of the Plinthaster or Sabogaster species, but it's really hard to tell. But it seems smooth, like the surface of the plates don't appear to display any granulation, and there's only a couple of possibilities. And if it was one of the latter, it could conceivably have been one of the new species that Okeanos collected during the last expedition. It'd be great to have a second individual in order to show variation and also occurrence, since we only have one data point for, or rather, sort of one specimen and 3 video observations of that species that